everyone, welcome to another video of inheritance and variation. We were discussing about genetic recombination and crossing over. These are just terminologies that you need to know. And if you would see in your textbook, a small paragraph is dedicated to these three terms. You should be clear about what is genetic recombination. And because of genetic recombination, because of crossing over, you will have two different types of progenies, that is the parental type progenies and the recombinant type of progenies that will be produced. Okay. So in this video, we are going to see the linkage two types, complete and incomplete, which was put forth by Morgan's experiment. Okay. Let's begin. Now, what is link or linkage? Okay, so the word itself is very simple. Linkage or linked genes. Okay, simply is what genes when they are on the same chromosome. Let's say two genes, you have A gene and you have B gene. Okay, both of them are on the same chromosome. And they are, if you will see by looking at the diagrammatic representation, they are very close to one another. They are very close to one another. So, chances of them coming together in the offspring, in the child, in the offspring, in the progeny is very, very high. So, they tend to be inherited together. Okay, because they are very close to one another. So, the crossing over, they getting separated and being expressed differently in different progenies is very less. They are very close together, and that is why they will travel also together. And both the traits of these genes will be expressed simultaneously in the progeny. Okay, so hence you can call them as linked genes. Fine, simple. Okay, so linkage and recombination. How this gives you linkage and I mean how recombination will come if the genes are linked. So there are two types of linkage: complete linkage or incomplete linkage, or another word is tightly linked or loosely linked. Then write completely over here, complete linkage or incomplete linkage. Okay, complete linkage or incomplete linkage. Right? So what happens in complete linkage or tightly linked? You see the distance between both the genes. And what is loosely linked or incomplete linked? You see a lot of distance between both the genes. Okay. So physical association of two or more genes on a chromosome is basically linkage. In general, not discussing about the types, but in general about the linkage, the distance between the two genes. That is, if there is no much distance, so this type will occur, and there is more distance between the two, this type will occur. So physical distance between two or more genes on a chromosome. Here we are taking only two genes. There will, there can be two or more genes that we can consider. So now when we talk about linked genes, the Mendel's independent assortment will not apply over here, because in Mendel's independent assortment, law of independent assortment, what would be saw that the genes were independent from one another. Okay, and let's say for example, A was independent and it could combine with any of the alleles of the another parent. Okay, but in this case, this A is quite it's very close to B. So both of them are dependent, sort of, or you can say they are close to one another. So they will go together wherever they are going in the progeny. They are expressed in the progeny. Okay, so these linked genes will not show independent assortment. Fine. This you need to know. And recombination will occur, generation of non-parental gene combination. So when we talk about loosely linked gene, their chances of their chance of getting expressed differently in the progeny is high, because there's a lot of distance between both the genes, right? Fine. So the non-parental gene combination you will see in this case. You will mostly see in this case. Okay. Whereas in this case, you will see the progenies will be looking more like of the parental type. Fine. Okay. Let's go into a bit more detail. So you have to be clear with the word linkage. Linkage is nothing but the distance between the two or more genes on the same chromosome. Okay, And linked genes usually will not show independent assortment. And depending upon what type of linkage it is, use complete link or incomplete link. Complete is tightly linked, there's no distance between the two, and loosely linked is more distance. They are located farther away on the same chromosome. So you will get recombinants in this case, and you will get parental types of progeny in this case. Fine. So Morgan carried out the experiment to find out complete and incomplete linkage. Now we have just understood the definition. How this concept was brought into existence, and how we know that okay, this phenomena also exists in the expression of genes. So Morgan carried out several dihybrid processes of the fruit fly to study sex-linked genes. Okay. So, so he crossed parents, that is one parent, one Drosophila, one fruit fly, which had gray color and normal wings, body normal wings, and the another parent was black color and no wings, vestigial wings, you can say. Right? So he chose these as one of example that we are discussing over here. Okay, to study the linkage pattern, and what did he found was that when he intercrossed their F1 progenies, okay, when he intercrossed their progenies F1, so the genes that were there, okay, gray and normal from one parent and the black and vestigial from the another, chances of them coming together in the phenotype, okay, chances of them coming together in the phenotype was very high. You can see the number 900, 909, 50 or 960. Black vestigial over here. Okay, that is approximately 950 over here again. And the recombinant genotypes was very less, roughly around 200 over here and 185 for the another type. Okay, so that is the recombinant means what? What did we saw from our previous understanding? Phenotypes which do not resemble the parents. That is gray and vestigial, okay, and black colored and normal wings. Right? So he found out that these two genes did not segregate it, they did not separate it only. Okay, they were coming together in mostly in you can say majority of them 900 900. Okay, majority of them represented the parents only, so the genes did not get separated independently, and the ratio that usually we get in the monohybrid cross of Mendel's monohybrid cross, that was not observed, that is 9 is to 3 is to 3 is to 1, that was not observed. Okay, so he found this out by carrying out this cross. Okay, and one more thing that we found out that most of these genes which were linked to one another, which were very tightly linked, which were close to one another, they were all mostly on the X chromosome. They were mostly on the X chromosome of the fruit fly, of this Drosophila fly. Okay, fine. So when two genes are situated on the same chromosome, proportion of the parental combinations was much higher as compared to the non-parental type. Okay, and this is because of the linkage. This is because of what? This is because of linkage. Okay, so Mendel did these process. One of the example that we just saw. You don't have to memorize this. This is not task exam. From this, you just have to know. Morgan experiment is just for understanding that from this experiment, the concept of linkage was put forth. Fine. Okay. So even thought. So this is the cartoon picture of Thomas and Morgan, and he says that genes located on the same chromosome, yes, will be inherited together. Independent assortment will not apply. Okay. So let's say white light, normal and gray color, and black and STG. So they are crossed together. You will see the progeny coming together. It is not like the monohybrid cross that you saw. One was two capital in the direct in the F2 generation. We saw two capital G once, and then hybrid condition we saw twice. Okay, and then the homozygous recessive condition one. So the ratio was 1:2. The genotypic ratio was 1:2:1, right? So that was not observed when Morgan carried out a process with respect to genes located on the same chromosome. That is when he did experiments on linked genes. Okay, fine. So this was the outcome, and this you need to know just the concept, that is what is linkage, what is complete linkage, and what is incomplete linkage. Okay, fine. So just a recap and conclusion that these tightly linked genes will have lower recombination rates. Mostly they will be representing parent type, and the loosely linked genes will show high recombination because their chances of getting separated and appearing independently in the progeny will be high. We just saw in the cross. Okay. So just a representation by looking at the gene, how they look like. This picture represents that. Right? Sorry. So you have the chromosome. This is linked. Gene 1 and gene 2 very close to it. Now this. Okay. So they are different process chromatids. In other stage, sister chromatids. Okay. Gene 1 and gene 2, you can see there's a quite a lot of difference between the two. So not really. Fine. So based on the linkage map, one can assume the farther the two genes are, the more likely a crossover will happen between them, and therefore the recombination progeny, the recombination frequency, or the number of recombinants that can be obtained will be here. Okay.